Ricin is a type of protein called a lectin, which is a specialized protein that likes to bind carbohydrates. It is found naturally in castor beans. The protein itself is composed of an A chain and a B chain. The B chain is responsible for binding to the surface of the cell. It will bind along carbohydrates that are attached to the phospholipid bilayer and other proteins. From this point, they're in a cytose, taken into the cell, which we'll talk about more in lecture, into a vesicle, backwards through the endosomal pathway. Eventually, the ricin is taken all the way back to the Golgi, backwards through the Golgi, and into the ER. Here, the A chain is released from the B chain. The A chain is released into the cytosol, while the B chain remains in the ER for now. Once in the cytosol, the ricin A chain can really jack up your ribosomes. It does this through a process called depurination that will take place at the ribosomal RNA component of the ribosome, so acting at the RNA. And here, what it's doing is basically destroying the purine nucleotides at critical places. This completely ruins the ribosome's function, which we know the ribosomes are necessary to make proteins. So if we don't have any proteins, we've got bad biscuits. So our goal in lab this week is to block ricin at the point of entry. So we'll be given 2 different drugs to test to see if we can block ricin's entry. To test which drug does this, we'll be using a special color-changing assay on cheek cells. So let's explore this protocol. To start the protocol, you'll be given a ricin-like molecule. It won't be ricin, it will be harmless, that will be modified. So here's our actual ricin mimic. You can see kind of the ghost of the A chain and the B chain. And it will be attached, attached to another enzyme called horseradish peroxidase. Horseradish peroxidase is really useful, and we use it a lot in cell biology for reasons we'll see in a moment. We may call this our HRP in the lab. So horseradish peroxidase is useful because you can give it a special substrate and it will convert it to a purple product. This purple product makes it a lot easier to see what's going on with the cells. Will also allow us to see where exactly our ricin mimic is hanging out. So let's look at 2 different scenarios. On the left, we'll be looking at what happens if we have an effective drug, and on the right, an ineffective drug. So again, you'll be given 2 drugs to test in lab, and you'll be determining which is the most effective. So first, we'll add our ricin HRP and our trial drug to the cheek cells. So you can see our ricin HRP. Here's our trial drug on the effective side. We'll add our ineffective drug on the ineffective side. So we'll allow these to all kind of incubate together over time. And what will be happening is we'll be allowing this drug to bind wherever it wants and allow the ricin HRP to bind where it wants it as well. So you can see here the drug is actually binding to those carbohydrate binding spots on the B chain. So we're actually blocking them. So it's not interested in the carbohydrates on the outside of our cell. In the case of the ineffective drug, we can see that we still get a lot of binding of this ricin to the carbohydrates on the outside of the cell, and the drug is not really doing anything. To continue with our protocol, we'll next add the substrate. So this is our colorless substrate, and we'll just allow the reaction to proceed. After the reaction has completed, we'll perform wash steps to ensure that anything that isn't bound to these cells, including substrate and additional ricin HRP, is washed away. If our ricin HRP is bound to the cell and thus could potentially enter it, we'll still be able to see the purple from this reaction. If we've successfully blocked ricin from binding to the outside of the cell in the first place, we shouldn't see anything. So you'll be given one of the two drugs to test in lab to determine which is more effective. So let's see how the protocol works.